The first module in the cardiovascular system is quite an extensive goal. It's all about the blood. The main objective of this module is to describe the characteristics, functions, and synthesis of the blood and blood cells. At the end, you should be able to answer the questions here. What is the function of blood? What is the composition of blood? What are the main types of blood cells and what do they do? How is blood made? How are red blood cells recycled? And how does the clotting process work? This figure is from the visual A&P book. The cardiovascular system has three main parts: the heart, which is the pump, the blood vessels, which is like the hoses, and the blood, the fluid in the hose. These work together to make sure all your tissues are getting the nutrients and oxygen they need and removing all the waste that they create. The main functions of blood are to transport gases, nutrients, hormones, wastes, etc. from the source to the targets, regulate the pH and ion composition of interstitial fluids because of the diffusion between blood and these spaces, as well as neutralizing via bicarbonate, clotting, preventing blood loss when injured, defending us against pathogens due to the white blood cells in blood, and we'll discuss this in even more detail when we get to the immune system, as well as stabilizing temperature. Because blood is fluid and most of that is water, which holds a lot of heat, and depending on the flow can retain or cause loss of that heat very rapidly. The main component of blood is plasma, which is mostly water. It also contains large plasma proteins and other solutes that may vary depending on what you had for dinner or your physiological status. The blood cells, red, white, and platelets, are also floating around in this plasma. Some numbers you ought to know. The average person has about 5 liters of blood. Men on average have a little bit more than this, and of course bigger people have more. You also have a bit more blood volume when it's warmer than when it is colder, so you have more blood in the summer. Red blood cells number varies with age and sex, but there's about 5 million per milliliter on average. So in a teaspoon of blood, if you can think of a regular spoon filled with blood, that's 5 milliliters, so you'd have 25 million blood cells in there. White blood cells are much less numerous, only 5 to 10,000 per mil, and also depends on your health status. One of the things we do is perform hematocrits to determine how many red blood cells you have, and about 45% of your blood should be red blood cells, or packed cell volume. So you take a small bit of blood, you put it in a tiny tube, and you centrifuge it, and the top section after you centrifuge it is going to be the plasma, this straw-colored stuff, and the bottom will be the red blood cells. The little line in the middle, the buffy coat, is where the white blood cells, and sometimes you can't even see this when you do your own hematocrit. The normal packed cell volume again is 45%, with the range being from 37 to 52, higher in men and lower in women. Plasma is the fluid that blood cells are in. It's 92% water. However, mostly we talk about the non-water components, because water is just water. The non-water components include proteins, hormones, waste like urea, uric acid, creatinine, carbon dioxide and water, or carbon dioxide and oxygen are the gases, other nutrients which may depend on what you had for dinner or lunch last, electrolytes, acids, bases, etc. There are homeostatic values or ranges for each of these, which can be seen on a complete blood count. So for example, your blood urea nitrogen should be 8 to 25 milligrams per 100 mils. While you don't need to remember each one of these numbers, you should know that doctors, nurses, etc. would look at all these numbers to help them diagnose diseases, which you may become more comfortable with after you're working in the field. There are 3 large plasma proteins in your blood. The important thing to remember here is that these proteins should only be found in the blood. You should never find them in lymph fluid or in urine or anywhere else. No, not in the interstitial fluid, nothing. Their main role is to stay in the blood and maintain osmolarity. Water will not rush in and out of tissues if the osmolarity stays about constant. If you need to remove osmosis, go back to your A&P1 notes. Besides maintaining osmolarity, each of the plasma proteins also do other things. Albumins, made in the liver, help carry free fatty acids and other particles around in the blood, so it's kind of a carrier protein. Globulins are important transporters of lipids and vitamins, and gamma globulins function as antibodies, which we'll talk about when we get to the immune system. Fibrinogen is a protein involved in the clotting process, which we're going to get to at the end of this presentation. Blood carries required nutrients to every part of the body and therefore contains all the nutrients we eat to all the parts. Most important is blood sugar. It's the nutrient of every tissue. It's required by the brain, and it should be at about 100 milligrams per deciliter, or 60 to 110. Amino acids are also carried in the blood to tissues, especially to the liver and muscle tissues, and they're required for protein synthesis. Lipids are also present in the blood. These seem to be of special interest to doctors because high levels of fats and cholesterols are linked with a higher risk of heart disease. And you can look up the numbers of these, but total cholesterol should be under 200 milligrams per deciliter. Blood gases like oxygen and carbon dioxide are also in the blood, of course. Your oxygen levels measured by a pulse ox thing, that little squeezy thing on your thumb, should be up there around 98, 99. They give you oxygen if you get much below 90. The wastes in the blood are removed by the kidney. These include urea, uric acid, other nitrogen sources. All of these can be toxic if allowed to build up. Electrolytes, again, any charged molecule is an electrolyte. Hopefully you remember that from A&P2. Perhaps the most common are sodium, potassium, and calcium. Play a big role in nerve impulse conduction, for example. The balance of these is extremely important to maintain for that reason. You don't want to have screwed up nerve impulses or muscle contraction. That would kill you. They also contribute to the osmolarity of the blood, and if the electrolyte is hydrogen ion, it can change the pH of the blood. So when you donate plasma, which you can get paid to do, you're donating all of these things. And you get your blood cells back, but they often use the other stuff for cell culture, research processes, analytics, that type of thing, to determine what kinds of things are in the blood. Alright, the three types of blood cells are red blood cells, known as erythrocytes, white blood cells, known as leukocytes, and thrombocytes, or the platelets. 99% of the blood cells are red blood cells. We have a lot of them. Their main function is to carry oxygen. White blood cells are going to play an important role in fighting infection, and platelets, even though they're only around for about 10 days, play an important role in clotting process. This beautiful picture, which was totally robbed from this copyrighted location, shows the red blood cells in red. The white blood cells, you can see one here in green, and the little yellow ones are the platelets. Shamelessly stolen and copyrighted by Dennis Kunkel. So we're going to start out and talk the longest, or the most, about erythrocytes. Their main function is to carry oxygen. You should know that. The shape of the red blood cell looks like a donut without the hole all the way through, and you have 5 million of these little biconcave discs chugging around your blood right now. Because of the shape, it allows them to have a larger surface area so oxygen can easily exchange, going between the capillaries and the tissues. They're also quite flexible and can squeeze through tiny capillaries. The average person, again, has 5 million per mil, and they're normally shaped like donuts unless you have the sickle cell mutation. This causes them to shape like sickles, and they get stuck in the capillaries and do not easily squeeze through. When oxygen is bound to red blood cells, they are brighter red, and without oxygen they are a little bit darker. So we can tell a little bit through our skin. The veins look a little bluer. It's because of the translucence of the skin, not because the blood is actually blue. But because of that, arteries are colored red and veins are colored blue on most models. Red blood cells are only around about 120 days before they're just too beat up to keep on working. They've been squeezed through too many tubes by then, and they have to be recycled. So here you can see the difference between a normal shaped red blood cell and a sickle shape. And if the sickle shape gets caught, which they're more likely to do, they're going to block blood flow to that tissue, and then that tissue is not going to get oxygen. That can trigger a severe pain response. Another good image of red blood cells, they look like little cinnamon candies, and kind of cute looking little things. Again, 5 million of them per mil. Red blood cells contain 280 million molecules of hemoglobin per cell. And because of that, they don't have nuclei and they don't have mitochondria because you want lots of room for hemoglobin. Hemoglobin is the protein that binds oxygen. And these 280 million hemoglobins each bind 4 oxygens, essentially resulting in more than 1 billion molecules of oxygen per red blood cell being carried around. One of the reasons there's so much hemoglobin is because there's a lot of room. There's not a lot of organelles. Because they don't have organelles, particularly nuclei and mitochondria, they can't do anything past glycolysis and they can't repair because they can't make any new proteins. This makes them lighter and it makes more room for the hemoglobin. If you're pumping around 5 million per mil and you have 5,000 mils of blood, even a little bit less each adds up to a lot of weight. When hemoglobin is bound to oxygen, it is called oxyhemoglobin. Very creative name there. Here is a structured molecule of heme, the protein which binds oxygen. Because I don't know if you can see here, but the oxygen is bound to iron, and if you don't have enough iron, you can't make enough heme, you can't make enough hemoglobin, you can't make enough red blood cells, you can't carry enough oxygen, you're tired. Okay, so people who have low iron can't carry enough oxygen. Sickle cells are mutated so their shape is slightly different. Instead of getting this ring, you get sticks, hence the sickle shape. Check out this YouTube for a good introduction to red blood cell synthesis and degradation. It's a really good one. You should take the time to listen to it if you haven't seen it in class. Red blood cells are continuously made because they're continuously being broken down. Remember, they only last 120 days. They are made in the red bone marrow, which is located in the spongy bone at the ends of the bones, the epiphyses. Particularly the flat bones, the pelvic bones, and the long bones are where red blood cell production occurs. As they're made, their immediate precursor is released. Those are reticulocytes into the blood at about 2 to 3 million per second, so you're kind of busy there. The hormone which causes red cell production is called erythropoietin, and when oxygen levels are low, erythropoietin is released from the kidney or the liver, goes to the bone marrow and says, kick it up, we need some more. You always have some erythropoietin being released, but more induces red blood cell synthesis. So here's a little flow chart of that. Again, low blood oxygen causes the liver or kidney to make erythropoietin, which travels through the blood, targets the red bone marrow, and says make some more, and more red blood cells are produced. So what do you need in order to make red blood cells? Well, you need the hormone, and in very few cases would a person be lacking that hormone. That's usually not the reason for low red blood cell production. However, you do need some. Second, you need folic acid. Folic acid and vitamin B12 both are required for synthesis, and lack of these would make people anemic. Folic acid is in your green leafy vegetables, and vitamin B12 is only found in animal products, so you need to eat the spinach and drink your milk, or eat meat. Vitamin B12 is absorbed in the intestine only when bound to intrinsic factors, so if for some reason your stomach doesn't make intrinsic factor, it will not bind vitamin B12 and absorption in the intestine does not occur later. People who are missing intrinsic factor have to get vitamin B12 shots and bypass the digestive system because vitamin B12 will just go right through you if you don't have intrinsic factor. And of course you need iron. Iron is required for the oxygen carrying ability of hemoglobin. Iron can be stored, although it is absorbed very slowly, it can sometimes be difficult to get from some foods. Unfortunately, it's easiest to get from eating liver or bloody meat, and if you're a vegetarian, that makes it pretty hard. If you do become anemic, it can take quite a while to restore your iron levels. It takes a long time to build back up. To help absorb iron, you need vitamin C. So when you're eating your egg in the morning, which does have a decent amount of iron, you should drink your orange juice with it. So what should a person check for when they're determining that you have anemia? Obviously, you're going to check your iron levels, but you should also maybe ask the patient about their vitamin B intake. Maybe measure their erythropoietin levels. Do they have intrinsic factor? The most common cause of anemia is low iron. Here are some different types of anemia. You don't need to know the different names of them, but there are different reasons for anemia. Okay, like I said earlier, red blood cells only live about 120 days, and the old ones get weary, they get raggedy, they get broken, squeezing through all those tiny capillaries all the time. So when the red blood cell gets old, as it moves through the liver or spleen, it is recycled. They rupture as they go through these organs, releasing their contents, and the hemoglobin is split between the heme and the globin, the two subunits of this protein. The globin can be further broken down to amino acids and recycled. But the heme, you got to take the iron off. When the iron is removed, which can be stored in the liver as bound to the protein ferritin or carried back to the bone marrow via transferrin, what you have left over after you take the iron off of that heme is a product called biliverdin. Biliverdin is a green color, hence the green color in the PowerPoint slide, and as it gets broken down further and metabolized, it becomes bilirubin, kind of a goldish yellow color, and eventually oxidizing to a brown pigment called biopigments, put into the bile, dumped into the intestine, and this is why your feces are brown. I'm sure you came to class today to learn about why your shit is brown, and there's the reason. Again, iron can be recycled and reused, but the old red blood cell production ends up mostly in the fecal matter. Some bilirubin may end up in your urine, also why urine is yellow. Here's a picture of some of those biopigments in the liver, or bilirubin granules, hence the color of liver, kind of that goldish brown. And when a newborn has jaundice, they have a buildup of bilirubin. Normally it goes through the liver and is excreted in the bile, but babies have to kind of catch up to this. They're sometimes builds up faster than the liver can handle it when they're first born. And a newborn baby, the bilirubin builds up can be dangerous, so you put them under UV light. UV light tends to break down bilirubin. Too much bilirubin can absolutely cause cerebral palsy, brain damage, etc. So they always want to check to make sure your baby's bilirubin levels aren't too high. The next type of blood cell that we're going to talk about are leukocytes. Leukocytes are the white blood cells, and there's about 5 to 10,000 per mil. Although this number can vary quite a bit depending on whether you're sick or not. High goes up when you're infected, for example. More common than to just count how many you have is to count how many of each kind you have. There are 5 types of white blood cells, and the neutrophils are the most common. More than 60% of your white blood cells are neutrophils, and that's the number you really want to look at. If you don't have enough of those, you can't fight anything off, and that's bad when your neutrophil count is too low. The word leukopenia means your white blood cell count is low, and viruses can cause our white blood cell count to decrease, which makes us more susceptible to other diseases. So when you get a fever, typhoid fever, or the flu, then you're more likely to catch pneumonia and other problems. Leukocytosis means your white blood cell count is high, and usually acute infections of bacteria can cause your white blood cell count to go up. So can loss of body fluids or vigorous exercise. Mostly though, again, the doctors, nurses, whatever, are going to take blood and they're going to do a complete blood count, a CBC, to differentiate between each different type, because one goes up, one goes down, that means different things. White blood cells do phagocytosis, which is engulfing and eating of debris, broken cells, bacteria, viruses, etc. They are the only cells that have ability to move out of the blood and into the tissues. That ability is called diapetasis, and that's what the figure there is showing. That white blood cell squeezing between the walls of the capillary and getting into the tissues. And they know where to go. A neutrophil will know where your splinter is, and they will all rush to the site of infection. Because of the chemicals released where there's injury, the neutrophils will home in on that. That's called chemotaxis. Again, one of the few cells that have the ability to do that. So the different types of white blood cells are listed here. 3 of them are granular, the background, or when you stain them, they have little grains in them that appear apparent in the staining procedure, where 2 of them are agranular, you don't really see the granules in the background. I don't care if you remember which one is which, but sometimes it helps you distinguish between them. And one of the things in lab is you'll be looking at these 5 different types of white blood cells and learning how to distinguish between them. The first one are neutrophils. Neutrophils are granulocytes. You can see in the background of that white blood cell there, the little granules in the back. Again, they're the most numerous or most common white blood cell. They are the frontline warriors, and without them you are in danger of becoming very sick very quickly. When you see the words neutropenic precautions on a patient's door, their neutrophil count is so low everyone must wash their hands, gown, put on gloves. Usually that patient can't receive fresh flowers, eat fresh fruit or vegetables, or even deli meats. Neutrophils are chemotaxic and phagocytic and are ready and waiting to fight off infection. Eosinophils are also granulocytes. They tend to go up when you have a parasitic worm infection and sometimes during allergic reactions. They are the only ones that stain red or pink, and the background again is granular. They usually have a bilobed nucleus and again are the only ones that are pink. Basophils are the last granulocyte. They go up during allergic reactions. They produce histamine and heparin, and histamine causes all the effects of an allergic reaction. Their background is blue granular. Sometimes they're called mast cells if they're in tissues. Monocytes are agranular, they're phagocytic, and they are the monsters. They have a distinctive large indented nucleus that reminds me slightly of a horseshoe or a baby butt. Like neutrophils, they play a major role in phagocytosis. Whoops, I'm sorry about that. I'll go back to that. They can live a long time. They're involved in the specific immune response. Sometimes they're called macrophages if they're in tissues because, again, they're very large and phagocytic. They can also help induce the antibody response during the immune response. And the last ones are the little ones, the lymphocytes. They're divided both into B and T lymphocytes. You can't tell that by looking at them in a microscope. But the B lymphocytes are going to be making your antibodies, and the T lymphocytes are become T killer cells. Although they're produced in bone marrow, they're transferred. The T lymphocytes mature in the thymus. That's where the T comes from. And you can find lots of these in the lymph system as well. They are specific to particular pathogens, which we'll again discuss when we get to the immune system. So which leukocyte is overactive in people with hay fever? That would be the basophils, and they secrete histamine, which causes most of the symptoms of allergies. White blood cells are also made in the bone marrow, just like red blood cells. There are hemocytoblasts or stem cells in your bone marrow that produce red, white, and platelets. Okay, so they're stimulated by slightly different chemicals. Erythropoietin stimulates red blood cell synthesis. Colony stimulating factors stimulate white blood cell synthesis. So here's your stem cell. You get the different whites as well as erythrocytes or red blood cells and platelets. And we can give people erythropoietin, we call it Procrit. It's what Lance Armstrong abused to make more red blood cells, or we can give Neulasta or Neupogen to induce white blood cell synthesis in patients. The last type of blood component are platelets. It's hard to call these cells because they're actually fragments of blood cells made from megakaryocytes. They're very tiny and are only around for about 10 days. You can see these little tiny blue flecks in the picture here. Those are platelets. They are required for the clotting process. They can release serotonin, which causes some contraction immediately of the nearby smooth muscles to help slow bleeding. And we'll talk about that in the next module. Here's an electron micrograph of a platelet. It looks like a chocolate chip cookie, which is kind of cool. The chocolate chips are the alpha and dense granules that contain a variety of mediators, and when activated they're released, and it causes everything to get sticky. So again, that helps with the clotting process. So hopefully you can tell what these blood cells are. Number 1, okay, and number 4 are both neutrophils. Number 2 is a monocyte. Number 3 is an eosinophil. Number 5 is a lymphocyte. Number 6 is a basophil. 7 is a platelet. And all the other ones in here are red blood cells. When a doctor orders a CBC, what does that include? That includes a complete blood cell count, so they're going to count each type of white blood cell as well as the reds and the platelets, and hopefully it will help them with a diagnosis for your disease. So don't forget, your online text and lesson are available to help you with this stuff. Don't be afraid to Google, and any textbook will have some information.